Hi everyone, my name is Stephen Kotiang, a graduate student at Wichita State University. Welcome to my poster presentation. The title of my presentation is Probabilistic Factor Graph Modeling and Analysis of Biological Networks. In this work, we formulated a probabilistic factor graph representation of biological networks and applied an inference algorithm to explore the global behavior of biological systems at steady state. This factor graph representation model treats biological networks as a cordon graph. This formulation can allow us to model gene regulatory relations mathematically. We then tested our methodology on gene expression data corresponding to two E. coli pathways and verified their consistency to the data. In biological perspective, our interest is to learn regulatory relations among genes in a network, as it determines the expression level of a gene from a set of genes interacting with it. Given a gene network, we transform it into an equivalent factor graph network as shown. A factor graph is a bipartite graph that associates variable nodes given in circles on one side of the graph and factor nodes given in squares on the other side of the graph. In our context, a factor node represents the regulatory function denoted Fi over a gene node Gi. In simpler terms, Fi denotes the probability that a gene takes a particular state with respect to an assignment from its parents. For the given gene network, the factor graph expresses the structure of the joint probability distribution factorization given by this equation. We then define the learning problem of F as selecting the maximum likelihood of data given the network model. We then performed marginal inference over the FGN model to determine the allowable stable states given the data. Since biological networks are cyclic in nature, we employed loopy belief propagation, a variant message passing algorithm for inference. We use belief propagation because they are simple, tractable, and a powerful message passing algorithm. In graphical models, the message passing algorithms work by iteratively computing messages at nodes and sharing their messages with the neighbors as shown in this figure. Here, lambda are messages from child to parent nodes, while mu are messages from parent to child nodes described by these two equations. Messages are defined as functions of the parents. After each iteration message update, the belief of the nodes are computed. This process is then repeated iteratively in time until a stable configuration is reached. The converged beliefs are the marginals of the variable nodes. We then tested our approach on E. coli SOS response network and acid resistance regulatory network. The acid resistance network is not shown here, but can be seen in our publication. For our model networks, we obtained uniformly normalized gene expression data from M3D database. The compendium data used on the E. coli networks contains 466 sample points. For our analysis, we discretize the continuous gene measurements into 2 and 3 logical states using mixtures of Gaussian distribution. We then applied our methodology to the data given network model. At convergence, for the E. coli SOS response network, the LBP predicted allowable stable states of the network for the 2 and 3 logical state discretization are summarized in this table and are compared against the experimental observed states, where table 1 shows the 2 states discretization and table 2 shows the 3 states discretization. Here, the marginals denote the probability of a gene variable being in a particular state. Similarly, for the acid resistance network, we show Pearson correlation plots between the inferred marginals and the observed states for both levels of discretization. These results show that our probabilistic model provides good approximation to the observed experimental data. As can be seen, the Pearson correlation coefficients are well above 0.90. We further verify the consistency of our networks to the gene expression data by comparing the performance of our model on these networks against their perturbed node networks. These two plots show the average correlation coefficients against increasing number of random norms. From the simulation, we observe that there is a general decrease in the correlation coefficients with increased number of randomized nodes in both levels of discretization. These results show that random GRNs are not consistent to the available gene expression data. In summary, we have explored the factor graph representation of biological networks where the converged marginals of the network describe the steady-state distribution of the biological pathway. Our findings reveal that the considered gene networks are strongly supported by the available biological data. Thank you.